We are the experiment team from Jefferson County, Louisville, Kentucky. This is Ben Bomarito, Cody Harrell, Alejandro Sid, Amber Walters, Miranda Fitz, oh my God. I'm Ashley Keister, and another one of our members, Jade Burton, could not be here with us. And we are testing the viability of microgravity, the viability of Lactobacillus GG on microgravity.
Our experiment is called the effect of microgravity on the viability of Lactobacillus GG. We did not begin with this experiment in mind. We had many other ideas, such as growing crystals, studying E. coli, fish eggs, and antibiotics. However, due to size constraints and materials lists, we decided to study Lactobacillus GG instead.
We chose this experiment after reading an article on Russian cosmonauts' gut flora. The human digestive system is home to trillions of microorganisms colonizing in the gut. These bacteria live together to help prevent diseases and are called gut flora. In this article, 20 cosmonauts that completed total 96 days in space were studied. Scientists measured the amount of gut flora levels before and after they completed the 11 missions. This research showed a decrease in the amount of gut flora levels. This made us curious to microgravity and had an effect of viability in gut flora, especially by Lactobacillus GG.
Lactobacillus GG is a probiotic. It's found in the gut flora. Its symbiotic relationship with humans help inhibit the growth of harmful bacteria. It also helps treat and prevent diarrhea. Lactobacillus has been proven to maintain a healthy intestinal tract and strengthen the body's natural defense. Lactobacillus is found in dairy products like yogurt and cheese.
Currently, NASA does not include probiotics in astronauts' diets. And as technology and space missions lengthen, NASA could very easily add these as meals or as a supplement. We use different forms of measurements to count our bacteria. One of those measurements was serial dilutions.
Serial dilution is the process by which we diluted bacteria by factors of 10 until the desired count of 9 million bacteria cells per milliliter was reached.
For example, we diluted the stock, pure bacteria, 25 times. Each dilution decreased the bacteria concentration to 1/10 what it was before. Through a series of equations, we determined that 10 to the negative 22nd was the closest to 9 million cells per milliliter.
9 million cells per mil was desirable because we did not want the solution to become saturated by growing bacteria. If this would have happened, bacteria would have ran out of food and space to grow.
When we received our sample from space, we placed 1 milliliter of our sample on Petri plates that contained an R2A broth. Also, we did the same thing with our control samples here on Earth.
We placed the bacteria on the plates because only live bacteria would grow, and that would allow us to count the number of colonies and compare the growth of the space sample to the Earth sample.
We did several series of serial dilutions and platings before we sent our sample while the sample was in space and when it returned so we could establish a general growth curve for the bacteria. At first, we had trouble getting the bacteria to grow on the plates due to the growth medium, R2A broth. Once we figured out the optimal R2A concentration, our bacteria began to grow and we were able to determine the growth curve.
We counted the colony forming units on Petri dishes. Typically, numbers between 30 and 300 are considered to be in the range where one's data is statistically correct. If the number of CFUs on our plate were greater than 300, we recorded it in our table as TNTC, or too numerous to count. We then plugged the number of colony-forming units into an equation that allowed us to determine the number of bacteria that was growing on the plates.
We used another method to count bacteria, but it was less beneficial in helping us gather our data.
A Spec 20 is a machine that shoots a ray of light through the sample. Based on the amount of light that passes through the sample, it helps us determine the amount of bacteria that is in the sample. When the light passes through the sample, we receive a number between 0 and 100. 0 meaning no light passes through, meaning the bacteria was bountiful, and 100 meaning the sample was clear like water, which meant the sample had no bacteria present.
Okay, so for our results, the graph is based on one-day results. We counted them, and on the bottom is the dilution factor, it goes from 0 to negative 30. Also on the right, that's our CFUs, or colony-forming units. The light blue line represents Earth, and the dark blue line represents space. The curve for space is very inconsistent. However, if negative 15 were higher, the curves would be very similar. Negative 15, therefore, is a possible outlier. The curve for Earth is much smoother, but counter to what we expected, which would be higher on the right and lower on the left. We do see a convergence at negative 20 and negative 25, which means that they were growing at the same rate. 2 days later, we counted the same plates, but found different results. There's more of a possible convergence starting closer to the pure stock rather than to the dilution. There are 3 points that are starting to converge now. The space curve increased dramatically, especially at negative 15, and that is where we see our third convergence. The curve for Earth is much different. Negative 10 is now a possible outlier, so if it were up higher, say right here, then the curve would have matched our expectation of higher on the right and lower on the left. There were, however, a few limitations to our project that may have affected our results.
It was very difficult to find a company that would supply Lactobacillus GG. This caused us to begin our experiment way later than we had first planned.
We based our experiment on research on Russian cosmonauts. The Russian team spent a total of 96 days in space, while our experiment only spent 15 in space. Therefore, the bacteria was not exposed to the same amount of time.
We used R2A broth as the growth medium for Lactobacillus. This is not an ideal medium. A broth containing lactose would have been a better choice. However, this was the only broth approved on the original materials list. We had a difficult time finding the ideal concentration of R2A for the Lactobacillus to grow. We made a 5%, 10%, and 15% concentration. After several platings, we concluded that the 15% R2A concentration was the best growth medium for lactose.
Another problem we ran into was inconsistency of our CFU counts. To help with this problem, we averaged multiple colony counts.
Some of our plates got contaminated during the process of finding our initial growth curve. This delayed our research because it forced us to start over. Getting plates poured, plating the bacteria, and waiting for it to grow was a week-long process. This also limited the amount of time we had to find the correct bacteria concentration to send to space.
If the experiment time were extended to 96 days to mock the cosmonaut research, our results could have differed in ways that could have changed our conclusion. Also, if we had put the bacteria under stress or under other factors to replicate the human body rather than growing in microgravity alone, our results would differ because our present results implied that the Lactobacillus GG actually grows better in microgravity rather than here on Earth. Up-to-date technology also would have made a difference in the accuracy of our results.
Thank you to the most important man in this room, Bo Lowry. Without you, we would not be standing here because you are the one that convinced our principal, Dr. Look, that in fact this experiment was a good idea and that we could go as far as anyone else. And here we are. Also, thank you to Colonel Vandermeer, Mrs. Herrick, Ms. Zipper, and Mr. Aper, and Mr. Harris for helping us with this project through school. Thank you to the sponsors, Kentucky Space, Fifth Third Bank, Wells Fargo, Louisville Water Company, Stars Restaurant, Murray's Heating and Air Conditioning, University of Louisville, Jefferson County Public Schools for providing us with the funding and materials for this important part of our lives. Any questions?
Thanks for a great presentation. Have you thought about, you mentioned that one possibility of, if you redo this experiment, would be to include some sort of stressor in the experiment. Have you considered that the fact that you used a non-optimal growth medium might have been a stressor and might also have affected the results?
Yes, we did not notice that the LB broth was put onto the revised list, and so the R2A broth actually was not the best broth for our bacteria, and so if we had actually changed it, then it would have shocked the bacteria, therefore it would have stressed them, and it would have mocked the human body, because the cosmonauts, we believe, were put under so much stress that it actually decreased their levels of the Lactobacillus GG, and that is why they had that much of a decrease over 96 days.
What about the fact that you had the experiment go in space for a long duration, and whether that could have been another stress factor with the limited resources that they had in very limited volume that everybody had to deal with, and whether that might have an effect on the growth of the bacteria, both in space and also on the ground?
Yes, since our experiment was only up in space for 14 days versus the cosmonaut total of 96 days, we believe that that has an effect on the growth that we noticed because they weren't exposed to the same amount of time in microgravity. And so we believe that if our experiment was up in space for 96 days like the Russian cosmonauts, we would have seen more accurate results, for lack of a better word, because they would have been exposed to the same conditions. Does that answer your question?
Yes, and one last question. A lot of people in their presentation have talked about the possibility of contamination from other types of bacteria. Did you notice anything like that in your sample, or did you look for it? Obviously, you get great results with the bacteria that you were using, but did you notice anything about possible contamination?
Yes, actually some of our plates did get contaminated, but that was because we did not have a very sterile environment. Our bacteria could have very well been contaminated during the dilution process because it was under a hood, but it wasn't closed. We had to go through a lot of gloves, I will say that. I don't know about the space, like all of your experiments. One thing that I was thinking about, on the shuttle, their environment has to be very sterile in order to keep the astronauts in pristine condition. So I don't know if that has anything to do with the fact that your experiments or ours were contaminated or not. So that could be a very possibility.
Right, I think that's something to consider definitely in the future is to make sure that we eliminate as many sources of contamination as possible. All right, thank you very much for a great presentation.